This is core component mock Holy Cross, mock 3, and we're up to question 5. Here we have a table, DNA and RNA, 3 differences between them, straightforward enough, I think so. And let's just keep it nice and simple. DNA has deoxyribose sugar, whereas RNA has ribose, and that's where the D and the R come from. And contains the base thymine. RNA is uracil. So nice, simple, straightforward answers we're trying to give there. Usually DNA is double-stranded, RNA is single-stranded. Some other things you could say there, but those are the big 3. Say those first. And we've got the classic Michelson and Stahl experiment using E. coli and isotopes of nitrogen. Hopefully you've revised this and done practice question on this. Lots of practice questions available. Explain why the density of the DNA in the new generation was intermediate. Well, they started off with that DNA, and then they added 14, and so these then split, and we could only make new strands from 14. And so Y was a new generation intermediate, because the new DNA had one normal strand, one heavy strand, and one heavy strand. And the 15N strands were templates, and N14 used to make complementary strand, and that's the semi-conservative replication. Which part of the DNA molecule contains the nitrogen? Well, it's not the phosphate bit and it's not the sugar bit, so it must be the other bit. It's the base. So they're sometimes called organic bases or nitrogenous bases. Cells are divided once more in the normal 14, and now we've got 2 bands. So if we go back to our diagram here, in the next generation, well, the same thing happens again. So these 2 split, and these 2 split. But we've only got 14 available to make complementary strands. I've done that one. Let's draw this again. There's our original DNA. This splits, and we've switched to 14, so there's only 14 available. Then these split, that splits into those two. That one splits into those two. And then to make the next strand, we're only supplying 14. And so on our diagram, this band here, the intermediate, is our those, and we've got two of those, and our normal DNA, well, we can't use the red pen at all, it's just our black 14 there. So what would be the proportion of the two types of DNA? Well, that's what we're looking at. So we've got two intermediates and two normals. So half and half. So 1 to 1, or 50% normal, 50% intermediate. Explain how you arrived at your answer. Well, it's just an explanation of this down here. So when the intermediate divides, well, we've got 15, a heavy strand, which we get another normal strand attaching to, but we've got a normal strand with another normal strand, and that's how we get our two different strands. So when intermediate DNA divides, the heavy strand gives intermediate DNA. The normal, it's not normal, is it? It's intermediate. The normal strand, yes, or the normal 14 strand gives normal DNA. So we're saying that this is an intermediate and this is a normal. Name the enzyme. The enzyme that makes DNA, that makes the DNA polymer. DNA polymerase. Most common right answer, RNA polymerase. Give an example of the practical application of this enzyme. Well, they use this at crime scenes where you recover a very small amount of DNA at a crime scene, and in order to test it and do genetic fingerprinting and things like that, you need to amplify or make more copies, and that's called PCR. So PCR, or an explanation, to make lots of copies of a small amount of DNA. You might not get that one, much more on that next year. But it shows how next year's topics will be intermingled with core topics. Explain how the sequence of nucleotides on the new strand of DNA is determined. Well, if we've got A's, T's, and G's down there, well, what are the rules for putting the new bases on? Well, that would have to be a T, that would have to be an A, and that would have to be a C. So that's just what you're trying to explain there. So by base pairing with the template strand, complementary base pairing or you give it examples, A's to T's, G's to C's. And then a little bit on the end, explain how a single alteration in the sequence might affect the organism's metabolism. Well, what's DNA for? DNA is the code for genes. Genes are the code, contains genes which are the codes for making proteins. The proteins make up the body, and proteins make up enzymes and so on. So you might make a faulty protein. So an enzyme reaction might not work properly, for example. So sequence of bases determine the sequence of amino acids on proteins or in proteins. Errors will give a different amino acid sequence, so the protein would not function. It might be a structural protein, or it might be an enzyme protein. And there's 14 marks, a classic bit of experiment that you should be able to relate in an exam.